Deputy Vice Chancellor Academic, Deputy Vice Chancellor Research, Innovation and Strategic Partnerships, the Registrar, University Librarian, Provost College of Medicine, Dean of Veterinary Medicine, Deans of other faculties, the postgraduate student of students, Directors of institutes and centers, Heads of departments, distinguished ladies and gentlemen. The rescheduled 421st in a series of University of Ibadan inaugural lectures will be delivered by Professor Funsho Olayemi of the Department of Veterinary Physiology and Biochemistry on behalf of the Faculty of Veterinary Medicine. Born on 30 July 1965 in Zaria, Kaduna State, to the family of the late Reverend Samuel Adepoju Olayemi and Mrs. Maria Alari Olayemi. On 30 July 1965, Professor Olayemi, who hails from Iragbiji in Borika local government area of Osun State, had his primary education at LEA Primary School, Kigo Road, Kaduna, from 1971 to 1977, and his secondary education in Baptist High School, Jos, Plateau State, from 1977 to 1982. He was offered admission to read veterinary medicine in this university in 1984, and he completed the program in 1990. He thereafter participated in the National Youth Service Corps scheme at the Federal University of Technology, Akure, in Ondo State, between 2nd of October 1990 and 1st October 1991. After a stint in the private sector between September 1992 and early 1994, he registered for the MSc Veterinary Physiology degree program of this university in 1994 and completed it in 1999. He later obtained the PhD degree in physiology from the Department of Physiology, College of Medicine of this same University of Ibadan in 2007. Professor Olayemi joined the Department of Veterinary Physiology, Biochemistry and Pharmacology, now Department of Veterinary Physiology and Biochemistry, as a lecturer grade 2 with effect from 1st February 1994 and was promoted lecturer 1 1st October 1998, senior lecturer 1st October 2001, reader 1st October 2007, and professor 1st October 2010. He has supervised many undergraduate and postgraduate students and is currently supervising 3 PhD theses. He has over 60 publications in both local and international journals to his credit. Within this university, Professor Olayemi has served in various capacities, including coordinator for veterinary undergraduate program from 1998 to 2007, and a postgraduate program coordinator from 2007 to 2017. Member of the Faculty Finance Committee 2015 to 2017, member Planning Committee for the 50th anniversary of the faculty, acting head, Department of Veterinary Physiology, Biochemistry and Pharmacology between 2009 and 2011. Head, Department of Veterinary Physiology, Biochemistry and Pharmacology between August 2013 and up to March 2017. And head, Department of Veterinary Physiology and Biochemistry from March 2017 up to 31st July 2017. Congressional representative in Senate between 1998 and 2000, and coming back again 2011 to 2013. Member of Senate as a professor since 2013. Member Senate Curriculum Committee 2015 to 2017. Consultant to the Veterinary Teaching Hospital. Pioneer member of the Servicom unit and member of the Publicity Subcommittee of the Organizing Committee for the hosting of the Inaugural Games in 2002. He is a 2-time recipient of the Senate Research Grant. Outside university, Professor Olayemi was a visiting lecturer to the Department of Physiology, College of Medicine, Olabisi Onabanjo University in Shagamu, Ogun State between 2007 and 2008, and visiting professor to the Department of Veterinary Physiology and Pharmacology, Federal University of Agriculture, Abeokuta, Ogun State, from 2012 to 2013. He served as external examiner at Ahmadu Bello University, Zaria, Federal University of Agriculture, Makurdi, Benue State, and the Federal University of Agriculture, Abeokuta, Ogun State. He is a member of many learning societies, including Physiological Society of Nigeria, Society of Medicinal Plants, Society of Toxicology, and American Society of Reproductive Medicine. Professor Olayemi is happily married to Mrs. Olasunbo Olayemi, and the marriage is blessed with children. I now call on Professor Olayemi to deliver his lecture titled The Axis of Life.
The Vice Chancellor, Deputy Vice Chancellor Administration, Deputy Vice Chancellor Academic, Deputy Vice Chancellor Research, Innovation and Strategic Partnerships, Registrar, Librarian, Provost of the College of Medicine, Dean of the Faculty of Veterinary Medicine, Dean of the Postgraduate School, Deans of other faculties, Dean of Students, distinguished ladies and gentlemen. Today's inaugural lecture is a testimony to the faithfulness of God, the giver of life, for his grace and mercy over my life. He gave his Son, my Lord and Savior, Jesus Christ, to redeem my soul and to give me the abundant life. The Bible says, when the Lord turned again the captivity of Zion, we are like them that dream. Then was our mouth filled with laughter and our tongue with singing. Then said they among the heathen, the Lord hath done great things for us, whereof we are glad. Psalms 126, verse 1 to 3. I'm a recipient of the grace and mercies of God, and it is with great pleasure I stand here to deliver the 424th inaugural lecture and the 22nd in the series for the 2016-2017 academic session on behalf of the Faculty of Veterinary Medicine. This inaugural lecture is the 38th from the Faculty of Veterinary Medicine and the first from the Department of Veterinary Physiology and Biochemistry. After the split from the Department of Veterinary Pharmacology and Toxicology, it is, however, the fifth from our mother department, the Department of Veterinary Physiology, Biochemistry, and Pharmacology. The table that has been projected are the past inaugural lecturers from the mother department. Professor M. Olokorum, a physiologist, gave the first on 9th May 1985. The second was given by Professor R.O.A. Arolo, a pharmacologist, on 4th April 1996. The third was given by Professor J. Oyewale, a physiologist, on 19 May 2011. And the fourth was given by Professor M.O. Abaton, a pharmacologist, 11th October 2012. The title of today's inaugural lecture is The Axis of Life. This first inaugural lecture from the Department of Veterinary Physiology and Biochemistry is coming off 77 days after it was to have been delivered. It was to have been delivered on 17 August 2017. 7 is recognized as a perfect number in the scriptures, which means today is most perfect to deliver this lecture. The Oxford Dictionary defines axis as a straight central part in a structure to which other parts are connected. The word axis are featured prominently in world affairs. On 27 September 1940, during the World War II, there was the formation of an axis referred to as the Axis powers, which was made up of Germany, Japan, and Italy. The Axis powers, also known as the Rome-Berlin-Tokyo axis, were the nations that fought against the Allied powers of Great Britain, United States, and the Soviet Union during the war. Again, on 29th January 2002, Mr. George W. Bush, the 43rd President of the United States of America, used the phrase axis of evil. We are all familiar with that phrase. During his State of the Union address, and it was often repeated throughout his tenure as president, to describe 3 countries that is administration accused of sponsoring terrorism and seeking weapons of mass destruction. In today's lecture, I will focus on another axis, the axis of life. Physiology, the study of life. Physiology has been described as the science of life. It is the study of the internal processes and functions that are associated with life in animals, plants, and microorganisms. Physiology highlights the mechanism of living, from the molecular basis of cell function to the integrated behavior of the whole body. It is derived from the Greek word physis, meaning nature, and logia, meaning study of, and was first used by the Greeks in 600 BC to describe the philosophical inquiry into the nature of things. Physiology explains how organs and systems within body function and how they combine their efforts to make conditions favorable for survival. The study of life has captured the imagination of philosophers and physicians from the early days of research. In a scholarly work in the late 18th century titled Physiological Researches Upon Life and Death, Javier Bichat wrote that life may be defined as an assembly of functions, those functions that offer resistance to death. The concept that life is opposition to decomposition has continued to receive attention from the pre-Socratic era through to Plato and Aristotle, and then to the 20th century with Charles Bell, Wilhelm Prof, Justus Liebig, and Charles Bernard. Physiology is the foundation upon which we build our knowledge of life, how to treat disease and how to cope with stresses imposed upon our bodies by the different environments. Physiological studies of normal biological function provide the basis for understanding the abnormal functions seen in animal and human diseases, which is pathophysiology, and for developing new methods for treating these diseases, which is translational research. The reproductive axis. There are different physiological axes that are recognized in the body, and some that are associated with the endocrine system include the hypothalamic pituitary adrenal axis, which is also referred to as the stress axis, the hypothalamic pituitary thyroid axis, which is also known as the metabolism axis, the hypothalamic pituitary liver axis, which is also known as growth axis, the hypothalamic pituitary mammary gland axis, which is also known as lactation axis, the hypothalamic pituitary gonadal axis, also known as reproductive axis. The reproductive axis, which is the focus of this lecture, is also known as hypothalamic pituitary gonadal axis, or the brain gonadal axis. That's in figure 1. It is a complex integrated system that is necessary for normal reproduction. The hypothalamus in the brain releases gonadotropin-releasing hormone, which is secreted in a pulsatile fashion from the preoptic and aqueous nuclei into the portal hypophyseal venous plexus. Gonadotropin-releasing hormone then stimulates the release of luteinizing hormone and follicle-stimulating hormone from the anterior pituitary into the systemic blood circulation. In the males, luteinizing hormone release is modulated by feedback of androgens at both the pituitary and hypothalamic levels. The release of FSH, or follicle-stimulating hormone, is regulated further by negative feedback of both inhibin and activin from the Sertoli cell or the testis. In the testis, LA stimulates testosterone production by Leydig cell, whereas FSH is crucial to the initiation and maintenance of spermatogenesis. Abnormalities anywhere in the reproductive axis have the potential for a negative impact on fertility in the male. Brood and Dishos, 2004. Reproductive axis and life. Mr. Vice Chancellor, sir. Scientists have established a positive correlation between the reproductive axis and longevity, especially in women. The reproductive cell cycle theory of aging, that the longer the reproductive axis is maintained in equilibrium, the longer an organism will live. The theory argues that the cellular changes are directed by reproductive hormones of the reproductive axis. Thus, the reproductive axis hormone will promote growth and development of the organism early in life in order to achieve reproduction. However, levels of the reproductive hormone then begin to reduce in men at around age 30, and abruptly in women when they reach menopause at around age 50. The dysregulation of reproductive axis, which occurs at menopause, lead to altered signaling to somatic and reproductive tissues, resulting in cellular dysfunction and or cell death. Atwood and Bowen, 2011, Yonka et al., 2013. Disease studies have shown that there are less cases of heart disease, stroke, dementia, and osteoporosis in women who reach menopause later, thus supporting the theory that the longer the reproductive axis is in balance, the less likely one is to develop age-related diseases. Henley et al., 2005. The reproductive axis in equilibrium will increase longevity. Life in the components of reproductive axis. Mr. Vice Chancellor, sir, the reproductive axis consists of the brain and the gonad, and as such, it is referred to as the brain-gonadal axis. The systemic blood circulation connects these two organs, as it is the case with hormone transport in the endocrine system. Blood transports the follicle-stimulating hormone and luteinizing hormone from the pituitary gland in the brain to the gonads, that is, testes in male and ovary in females. The three components of the reproductive axis mentioned above are critical to life and survival of the animal. And in this lecture, they are referred to as the axis of life. The brain has long been regarded as the seat of consciousness, emotion, and holds the most central vital functions in the body. The importance of brain to life is encapsulated in the brain life theory that was postulated by Goldring in 1985, which says that whenever a functioning human brain is present, a human being is alive. Agnion of Croton, a pre-Socratic physician philosopher, introduced the concept of mind and soul being located in the brain. Agnion argued that the brain is the seat of human intelligence, Celestia 2012. The brain occupies a unique place in the body of humans, and the knowledge gap that needs to be filled in the field of neuroscience is so wide, which was probably what prompted Mr. George A.W. Bush, the 41st President of the United States of America, to issue the 6158 proclamation on 17 July 1990, in which he designated 1990 to year 2000 as the decade of the brain. It is heartwarming to note that giant strides were made by neuroscientists during the decade. The important role of male and female gonads in the production of gametes, that is, spermatozoa in the male and ovum in the female, ensures that at fertilization, life is propagated. The essence of reproductive processes is to produce an offspring, which is a new life, and this ensures the continuation of species. Semen comes from Latin word semen, which means seed. It was once thought that semen was like a seed that grew into a baby after being planted inside a woman's body. We now know that semen contains the sperm cell which fertilizes the ovum to give rise to zygote, which eventually results in the offspring, that is a new life. Blood is a connective tissue in fluid form, and it is regarded as the fluid of life because it carries oxygen from the lungs to all parts of the body and carbon dioxide from all parts of the body to the lungs. The Holy Bible says, "But the life of the flesh is in the blood, and I've given it to you upon the altar to make an atonement for your souls. For it is the blood that maketh an atonement for the soul." Leviticus 17 verse 11. It was the discovery by William Harvey, which he reported in 1628, that showed that circulation of blood maintained the biological life by supplying nourishment to all parts of the body and also carries away its waste. There must be sufficient blood flow around the body, or else body function deteriorates, and this leads to death. My research efforts for the past 23 years are focused on these three different systems of the reproductive axis: the brain, the male reproductive system, and blood in the systemic circulation. In this inaugural lecture, I will highlight my contributions under the following subtitles: the male gonad in the axis of life, blood in the axis of life, the brain in the axis of life. The male gonad, that is testis, in the axis of life. In the male, the reproductive axis is also known as the brain testicular axis. The testis is the male gonad where the gametes are produced. The male reproductive system aims at producing viable male gametes, that is spermatozoa, that fertilize the female gamete, that is the ovum. Male gametes are the smallest human cell, that is smallest cells in the human being. The sperm, which is only 5 micrometers by 3 micrometers in size, excluding the sperm tail. The female ovum is, on the other hand, the largest human cells, which is 120 micrometers in diameter. That is about 24 times the size of a sperm cell. Fertility is the natural capability to produce offspring, and male fertility is very important in the reproductive processes of man and animal. Reproductive axis and male infertility. Infertility is the inability of humans, animals, or plants to reproduce by natural means. Infertility in humans is defined as 1 year of unwanted non-conception with unprotected intercourse in the fertile phase of the menstrual cycles. Evers, 2002. The main cause of male infertility is low semen quality, and as such, a normal semen sample in man should have a volume of 1.5 to 5 mils, and with greater than 20 million sperm cells per male. The number of abnormal sperm should be less than 40%, with greater than 30% of sperm sample demonstrating proper motility. An estimated 40 to 90% of male infertility is due to deficient sperm production of indefinable origin. Sinclair, 2000. A large proportion of infertile male fail to conceive because of lack of sperm, which is known as azoospermia, too little sperm, oligozoospermia, abnormal sperm morphology, which is teratozoospermia, insufficient sperm motility, or adenozoospermia, Frank, 2003. Influence of medicinal plants on the reproductive axis. The interest of my research group was drawn to the anti-fertility effects of medicinal plants due to the fact that medicinal plants play a crucial role in the healthcare needs of people around the world, especially in the developing countries. Becalo et al., 2009. About 80% of the population of most developing countries still depend on the use of traditional medicines derived from plants. Salo, 2016. The anti-fertility effects of many medicinal plants have been reported to be true disrupting of the hormonal regulation of spermatogenesis and steroidogenesis in the reproductive axis. Many medicinal plant products have been reported to target lady cell and hinder their endocrine function. That's it, being projected. Most of the medicinal plants studied impair steroidogenesis by targeting the enzymes involved in the process at the level of lady cell or at the level at the hypothalamic pituitary gonadal loop. Ubewu et al., 2011. The nexus between antimicrobial and male infertility. It has been reported that many antimicrobial agents possess male antifertility activity in varying degrees. Chloroquine, quinine, and quinacrine were reported to inhibit lady cell steroidogenesis and thereby induce infertility in male. Schlengel et al., 1991. Adeyeko and Dada, 1998. Yoshinobu et al., 2005. Antibiotics such as erythromycin, tylosin, ampicillin, and clozacillin were shown to have male antifertility effect. Mercer et al., 1993. Raja et al., 2006. Mr. Vice Chancellor, sir. Interestingly, many anti-malaria and antibacterial medicinal plants extracts have also been implicated in male infertility. For example, Quasar amaria, which is highly potent against chloroquine-resistant Plasmodium falciparum, produced significant reduction in epidermal sperm counts, serum level of testosterone, luteinizing hormone, and follicle-stimulating hormone in the male rat. Raji and Bolarinwa, 1997, Pavin et al., 2003. Astona boni, a tropical plant reputed in traditional medicine to have anti-malaria activity, Olajide et al., 2000, was reported to cause duration and dose-dependent reduction in sperm characteristics, Raji et al., 2005. Azadirachta indica, which is popularly known as the dogoraro, is a medicinal plant with potent anti-plasmodium activities, Billy, 1986. It was reported to induce male anti-fertility effects, Joshi 1996, Raji et al. 2003. My contributions. Male anti-fertility effects of Netis ferruginea. My research group carried out series of studies on Netis ferruginea decando in male rats, which was reported to have antiplasmodial activities, Bregaloo and Wright, 2005, to determine if it also has male anti-fertility effects. Netis ferruginea is in the family Conariaceae. Phytochemistry revealed that alkaloid and flavonoids are present in leaf extracts. It is known as Omoaja or Goingoi in Yoruba. We first carried out the safety assessment of the methanol root extract of the plant, and we established that the dosage of up to 2,000 mg per kilogram body weight was not lethal to the experimental rats, and the pathological values were not adversely affected. Olayemi, Raji, Adekoke, and Olayemi, 2010. That's in table 1. Thereafter, the effects of methanol root extract of Netis ferruginea on reproductive indices were studied in male rats. The animals were treated daily with 500 mg per kilogram body weight for 5, 30, and 60 days. There was a group of rats which was allowed 30 days to recover after 6 days treatment. There was a significant reduction in sperm count, viability, motility, as well as weight of the testes in all the different duration treatment groups. That's the figures 10A and 10B. Plasma testosterone level was severely reduced only after 5 days treatment period, figure 11. After the 30 days recovery period, the sperm motility and viability were restored, but the sperm counts and sperm morphology were not fully restored. The study showed that Netis ferruginea possesses male anti-fertility activities. Olayemi, Raji, Adekoke, and Olayemi, 2011. We were able to purify the crude extract of Netis ferruginea into fractions using column chromatography and thin layer chromatography. With the aid of nuclear magnetic resonance and mass spectrophotometry, we isolated a quinolisdine alkaloid as a bioactive compound from the root extract of Netis ferruginea. That's in figure 12. The pure compound was then given to male whistle rats at varying dosages of 0.1, 1, and 2 mg per kilogram body weight. It caused significant reduction in sperm count, motility, viability, and morphology, figures 13A and 13B. The plasma levels of testosterone, luteinizing hormone, and follicle-stimulating hormone in male rats were also reduced significantly by the pure compounds of Netis ferruginea, figures 13C and 13D. This shows that the mechanism of action of Netis ferruginea was by disrupting the reproductive axis, Olayemi and Raji, 2011. The fertility index was confirmed using a mating experiment. It was observed that female rats that were mated with male rats treated with the biotic compound of Netis ferruginea had similarly lower litter size than the control group, Olayemi and Raji, 2011. That's figure 14. The anti-fertility effects produced by the pure compounds of Netis ferruginea were, however, reversible after 30 days of withdrawal of the pure compound. The altered sperm characteristics reverted to the pre-treatment values. Mr. Vice Chancellor, sir, apart from publishing our findings on the infertility caused by Netis ferruginea in both local and international journals, these findings were also widely disseminated by a report in the Tribune newspaper of 1st March 2012 with the title, Infertility, Reason Men Should Not Abuse Herbs. Male anti-fertility effects of Wateria indica. My team then studied another Nigerian medicinal plant for possible anti-fertility activities. Wateria indica, which is also known as sleeping money, belongs to the family Staphylococcus. It is widespread in West Africa sub-region. It is known as Eweko in Yoruba, and the aqueous extract of the root and stem is used to treat syphilis. Wateria indica has also been reported to have anti-malaria and antibacterial
effects. Male whistle rat were administered varying dosages of the aqueous leaf extract of Bacteria indica. There were significant decreases in sperm count, motility, and viability, but significant increase in the abnormal sperm cell of treated whistle rat, Bashiru and Olayemi 2012. That's in table 2. The purification of the crude extract of this plant is ongoing in our laboratory so as to establish the active principle responsible for male anti-fertility. Male anti-fertility effect of Modica charantia. Excuse me. Modica charantia, which is called ejire in Yoruba, is popularly known as bitter melon. It is a medicinal plant in the family Cucurbitaceae. This plant has been reported as being one of the most promising in the treatment of diabetes, NAHAS, and MOHA, 2009. We investigated the possible reproductive effects of the leaf decoction of Modica charantia in male rats. At a dosage of 450 mg per kilogram body weight, the plant caused a significant reduction in sperm motility, concentration, and viability in male rats. Odewusi et al. 2010. Male fertility enhancing effect of Arconia codifolia. Mr. Vice Chancellor, after establishing the anti-fertility effect of some medicinal plants aforementioned, the attention of my research group shifted to the study of a plant which is believed to enhance male fertility. This study has become very necessary considering the current prevalence of male infertility in our society. Arconia codifolia, which is called a pie in Yoruba, is famous among rural dwellers due to its effectiveness in enhancing male fertility and the treatment of venereal diseases. The result of our study showed that the methanol leaf extract of Arconia codifolia has male fertility enhancing effect. Ajibade and Olayemi 2015. The leaf extract caused significant increases in testicular weight, sperm count, motility, and serum testosterone level at 200 and 400 mg per kilogram body weight. Table 3 and figure 18. Efforts are ongoing at purifying the crude extract so as to isolate the pure compounds that are responsible for the enhancement of male fertility. Blood in the axis of life. Mr. Vice Chancellor, I mentioned earlier that the reproductive axis, the systemic blood circulation, conveys gonadotropins from the pituitary gland in the brain to the gonads. Systemic blood circulation is the second component of the reproductive axis. Apart from this very important function of transporting luteinizing hormone and follicle-stimulating hormone from the brain to the gonads, blood is also involved in the mechanism of penile erection, which is necessary in copulation for fertilization to take place. Protrusion of the highly vascular penis is by the engorgement of the sinuses with blood, and for this reason, adequate blood supply to the male reproductive system is essential in reproductive processes of man and animals. Marisa reported the hematinic effect of medicinal plants and the baseline hematological values in animals in our tropical environment. Hematinic is an agent that stimulates the production of red blood cells or increase the amount of hemoglobin in the blood. Marisa team reported the hematinic effect of ethanol extract of Troboma cacao stem bark, that is the popular cocoa tree stem bark. Tea cacao belongs to the family Malvaceae and it is reported to have high levels of polyphenols and potent antioxidant activities. Prof et al. 2002. Erythrocyte values, which were similarly reduced when rats were treated with phenadrazine, were fully restored to the normal values with the oral administration of the stem bark extract tea cacao. Treatment with tea cacao at 500 or 1000 mg per kilogram body weight similarly increased the PCV, HB, and RBC value in the rat. Shagodoyi, Olayemi, Drotoye, and Ajibade 2016. That's in table 4. Furthermore, we reported that methanol leaf extract of Arconia codifolia, which I earlier mentioned as enhancing male fertility, did not have any toxic effect on the blood profiles of rats after treatment for 30 days with up to 400 mg per kilogram body weight in rats. Ajibade and Olayemi 2015. That's in table 5. This shows that Arconia codifolia is a good candidate for development into a male fertility drug because of the lack of toxic effect. Hematology of domestic and wild animals in Nigeria. Mr. Vice Chancellor, apart from studying the hematinic effect of medicinal plants, I have also determined the baseline hematological values of animals in our environment. I was employed as a lecturer in veterinary physiology in February 1994. And for the first 10 years, my research focused on the hematology of some domestic and wild animals in our humid tropical environment. Early in my career, we observed that the baseline hematological values of our indigenous breeds of domestic and wild animals have not been well documented. There was the urgent need to determine the baseline hematological parameters for our indigenous breeds of animals. The baseline hematological values were therefore determined by my team for the first time in many local breeds of animals. The influence of gender and several other physiological factors on hematological parameters were elucidated by our research efforts. The studies generated significant findings that are now being cited internationally. One of such was the citing of my publications in the books written by my friend Professor Chester Glonsky, a professor of comparative hematology at the Jacobs School of Medicine and Biomedical Science, University of Buffalo, New York, Mr. Vice Chancellor. In appreciation of our contributions to his book, Professor Glonsky gave copies of his textbooks to our faculty and university libraries. My contribution on the hematology of domestic animals, Nigerian dog. We carried out studies on the blood profiles of Nigerian dog, Anas matrigos. We observed that the erythrocyte, glucoside, and plasma biochemical values were similar in the male and female Nigerian dog. Olayemi, Arolo, Saba, and Famakide, 2002, Table 6 and 7. We also reported similar leukocyte values in the young and adult Nigerian dog, Olayemi, Oyewale, Rahman, and Omolewa, 2003. However, the young dog has significantly higher plasma aspartate aminotransferase and alanine aminotransferase values than that of the adult, but the values of alkaline phosphatase and plasma electrolytes were similar in the young and adult Nigerian dog, Olayemi, Oyewale, and Omolewa, 2002. Nigerian turkey. There was a dearth of information on the hematology of Nigerian local turkey, and this informed our studies on the hematological values of this bird. Previous studies revealed the hematological parameters of the white England turkey in the hot, humid tropical environment, Makinde and Fatumbi, 1985, Oyewale and Ajibade, 1990. Our study observed that the erythrocyte and leukocyte values were similar in the male and female Nigerian turkey, Olayemi and Ojo, 2007, Table 8. However, the RBC, HB, MCH, MCAC, WBC values and erythrocyte osmotic fragility were significantly higher in the young than adult turkey, Aziz Olayemi Olaraju, 2011. Nigerian cattle. We carried out extensive studies on the hematology of white Fulani cattle. The effect of management practice, gender, and breed on the blood values of the white Fulani cattle were investigated. We observed that the intensively managed white Fulani cattle at the International Institute of Tropical Agriculture in Ibadan, Oyo State, has significantly higher PCV, RBC, WBC values, but significantly lower erythrocyte osmotic fragility than the extensively managed cattle by the nomadic herdsmen, Olayemi Oyewale, 2002, tables 9 and 10. We also reported that the plasma protein and albumin concentration were significantly higher in the intensively reared white Fulani cattle, Olayemi Oyewale Fajimi, 2001. That's table 11. These studies underscored the need to ascertain the management regime of white Fulani cattle in interpreting the hematological values. We also reported that the hematological values of white Fulani cattle were significantly different from other breeds of cattle in Nigeria, such as the Indama cattle, Kure cattle, and Gudale cattle. Nigerian dog. The need for a reliable reference hematological data necessitated this study, in which we determined the influence of gender, age, and breed on the blood profiles of the Nigerian dog. We reported that although the erythrocyte values were similar in the male and female Nigerian dog, the erythrocyte osmotic fragility was significantly higher in the female than the male Nigerian dog, Olayemi et al. 2009. We also reported that the erythrocyte values of the German Shepherd and the Nigerian dog were similar, but the white blood cell and neutrophil counts were significantly higher in the Nigerian dog, Olayemi and the Gahon, 2009. That's in table 12. The erythrocyte osmotic fragility of the German Shepherd was, however, higher than that of the Nigerian dog, Olayemi and Olayemi 2016. We also reported that the blood values of Nigerian dog were similar to those of the Rottweiler dog Adebiyi, Ajayi, and Olayemi 2014. Hematology of some wild animals found in Nigeria. African fruit bat. In 2014, the African fruit bat, Edolon helping, was reported as the presumptive reservoir host of Ebola virus. Mr. Vice Chancellor, long before the 2014 outbreak of Ebola virus disease in West African sub-region, we reported for the first time the hematology of African fruit bat, Olayemi et al. 2006, table 13. We observed that the white blood cell value of the African fruit bat was significantly higher than those of the temperate breeds of bat, Myotis, and Myotis elegans. The significantly higher WBC value in the African fruit bat may be the reason why they are one of the successful reservoir hosts of Ebola virus, African giant rat. We carried out a series of studies on the hematology of African giant rat, Chrysomelis gamenus waterhouse. The white blood cell counts and the erythrocyte motif fragility were significantly higher in the female than the male African giant rat. But all the other hematological parameters were similar in the two sexes. Oyiwale, Olayemi, 1998, table 14. We also reported that the plasma concentration of electrolytes, enzyme, protein, and metabolites were similar in the male and female African giant rat. Oyiwale, Olayemi, and Ogunsanmi, 1998. We later established that the erythrocyte values were not influenced by estrous cycle in female African giant rat, but the white blood cell count was significantly different at the different phases of the estrous cycle, and Olayemi 2000. We also documented the seasonal and diurnal variations in the blood profiles of the African giant rat. We thereafter reported that except the white blood cell count that was significantly higher in the African giant rat, the blood values of the African giant rat and African grass cutter were similar. Olayemi, Insien, and Ozegbe, 2006. The brain in the axis of life. The brain is an important component of the reproductive axis. The hypothalamus, which produces and secretes luteinizing hormone-releasing hormone, and the pituitary gland, which produces and secretes luteinizing hormone and follicle-stimulating hormone, are located in the brain. The LH and FSH are transported in the blood to the gonads. It was not until when Professor J. Olopade, the current dean of my faculty, and I completed the supervision of my former PhD student, Dr. Olamide Adebiyi, in the field of neuroscience, that I realized that my 23-year career as a researcher has been along the reproductive axis, which I describe as the axis of life. Neuroprotective effects of Guera caprifolia against vanadium-induced toxicity. The plant, Guera caprifolia, belongs to the family Tilaceae. It was reported to have antioxidant properties. Goyal, 2012. There have been reports that temperate plant species of the genus Guera elicited varying CNS activities. 4 and score, 2004. Chamon et al. 2014. However, the likely neuroprotective activity of Guera caprifolia, which is found in Nigeria, has not been investigated. We therefore carried out a series of experiments to establish the neuroprotective role of crude and pure extract of Guera caprifolia on vanadium-induced toxicity in mice. The environmental pollution caused by vanadium through glass flaring has assumed a worrisome proportion. In the 70s, the whole world was emitting 35,000 tons. But as of 2011, Nigeria alone is emitting almost double that amount, 65,000 tons. And Nigeria is ranked second in the world. But when one consider that it's only the Niger Delta region that is producing this vanadium through glass flaring, it is definitely the worst polluter in the world. We designed a study to investigate the effect of ethanol leaf extract of Guera caprifolia on the behavioral alterations following acute vanadium toxicity in mice. Guera caprifolia, given at 200 milligram per kilogram body weight, significantly reduced the number of grooming, stretch, attempt posture, and freezing time that were significantly increased in the group that received only vanadium. And Guera caprifolia also enhanced the vestibular functions. Adebi, Olopade, and Olayemi 2016. That's in table 15. We then proceeded to evaluate the free radical scavenging activity of ethanol extract of leaf and stem of Guaiac capifolia using various in vitro models. We used the DPPH quenching assay, ABTS cation declaration test, FRAP, and TBAS assays. The result of this study revealed that Guaiac capifolia has a high radical scavenging activity in the various radical systems. The excellent antioxidant activity of Guaiac capifolia extract may be due to the high level of flavonoids and phenols in the plant. Adebi, Olayemi, Nihol, and Guanzi 2016. Figures 29 to 32. Nuclear magnetic resonance spectroscopy revealed that beta spinosterol, diabetes folate, beta cytosterol, benzoic acid butyl ester, and stigmasterol were the 5 bioactive compounds that were isolated from the leaf and stem extracts of Guaiac capifolia. Only 2 of these, that is, beta cytosterol and stigmasterol, crossed the blood-brain barrier. They exhibited potent antioxidant and neuroprotective activities. They are therefore potential candidates in the treatment of vanadium neurotoxicity. Conclusion. Mr. Vice Chancellor, I've shown in this inaugural lecture that physiology is the study of life, and I highlighted the importance of each of the systems involved in the reproductive axis to the life of the animal. The brain and the gonad, which constitute this axis, are linked by the systemic blood circulation. My research efforts have touched these different components of the reproductive axis. I have shown in this lecture our findings that medicinal plants, Lettuce virginia, Wateria indica, Amabolica charantia, have male anti-fertility effects in rats. I also mentioned in this lecture our findings on the male fertility enhancing activities of methanol leaf extract of Aconia corifolia in rats. It was shown in this lecture the hematignic effect of tribunal cacao stem bark, and I described our significant contribution to the field of hematology in the different species of domestic and wild animals that are found in this country. The neuroprotective role of ethanol leaf extract of Guaiac capifolia in vanadium-induced toxicity in mice was also revealed in this lecture. These findings of our research efforts presented in this lecture describe the contributions of my research team in protecting the reproductive axis and ensuring it remains the axis of life. Recommendations. Mr. Vice Chancellor, sir, I wish to make the following recommendations. It is established that therapy with medicinal plants is a viable alternative to orthodox medicines. Their acceptance is due to the fact that they are excellent antioxidants and they possess limited side effects that accompany their consumption. It is however recommended that they should be administered with caution, and indiscriminate consumption of medicinal plants should be avoided, as it has been shown in this lecture that some medicinal plants with antimicrobial activities are implicated in male infertility. The relevant regulatory bodies should ensure the standardization of herbal drugs, and there should be quality control in the commercial production of these drugs. 2. The improved blood profiles of the intensively managed white Fulani cattle that are revealed in this lecture highlights the advantage of rearing cattle intensively on ranches over the nomadic grazing of these cattle. The extensive migratory grazing of cattle by the Fulani herdsmen should therefore be discouraged by the government. 3. This lecture focuses on the reproductive axis, and each component of the axis is very vital to the life of man and animal. There is also an axis in this university which is made up of the management, which are likened to the brain of the reproductive axis, the teaching and non-teaching staff, which are likened to the blood in the circulatory system of the reproductive axis, and the student population, which are likened to the gamete production in the reproductive axis. Each component of this axis in the university is very important to the life and survival of this university. And just as disruption of the reproductive axis leads to infertility, disruption to this university axis at any level will lead to inefficiency and low productivity of the university system. It was highlighted in this lecture the importance of protecting the reproductive axis. Likewise, the diversity axis should be protected from hostile factors, and this will ensure that we produce students with knowledge and sound acknowledgements. My deepest and most profound gratitude goes to the Almighty God, who has been a very present help for me in my journey to the peak of my career. The Lord is truly faithful, and I appreciate his unfathomable love to me. I'm blessed with wonderful and loving parents, and I wish to thank my father, the late Reverend Samuel Adepoju Olayemi, for giving me quality Christian upbringing and instilling discipline and godly values in me. He saw into the future and strongly supported my studying veterinary medicine despite some opposition to that. My mother, Mrs. Maria Alari Olayemi, she's here seated today, gave up a lot to ensure that I had quality education, and she has always been very supportive. She instilled in me the virtue of honesty and diligence. My mother sacrificed her comfort in ensuring that we all had our tertiary education after our father was called to glory. I'm truly grateful to my siblings, Mr. Femi Olayemi, Mrs. Olayemi Ayedun, Mr. Akitude Olayemi,